name is Dr. Nina Steele. I am an assistant scientist here at the Henry Ford Pancreatic Cancer Center, which is where we are right now. My lab is focused on understanding how different types of cells in pancreatic cancer communicate with one another. In particular, we have two focuses in the lab. The first focus is understanding developmental pathways that are important in the formation of an embryo and how those gene pathways get reactivated inappropriately in the context of cancer itself. The second focus in the lab is understanding that this disease does not actually influence all racial groups equally. Pancreatic cancer is 20% more likely to develop in individuals who are Black or African American. So we have a really important obligation here at Henry Ford, given that our patient demographics are around 25 to 35% of patients that are diagnosed with pancreatic cancer here are Black or African American. We are able to utilize those tissues to study these racial disparities in this field. There is a revolution in a field that is very close to my heart, which is big data. So the genomic resources or gene expression information that we can gather from each individual patient is really astounding. From one patient tumor, we can look at expression of tens of thousands of genes in millions of cells. And so with each person contributing their patient tissue to research, this all compounds and grows together. So over the last 2 years, my lab has spent significant efforts in compiling a human atlas of the pancreas. So far, we have been able to gather information, gene expression information, from over 1 million cells, which is representing 229 patients, which is a very large atlas collection of data. So I think that by using artificial intelligence and deep mining learning algorithms, we can leverage this big data information to try to answer a couple of key questions. How and when does pancreatic cancer arise in patients? Why are some individuals resistant to certain treatments but not others? In other words, can we define which patients will have specific vulnerabilities to drugs that we can use to try to kill the cancer cells? So these are some of the aspects of hope that I think are coming in the future, but it really requires the synthesis and the teamwork to bring all of this big data into one place and help researchers to understand the complexity of this disease. Like many other cancer researchers in the world, I was influenced by the diagnosis of two of my immediate family members when I was in graduate school. So I actually didn't start out studying cancer. I was studying a bacteria called Helicobacter pylori, which affects the stomach lining, causes inflammation, but can also lead to increased risk of stomach cancer. But when I was a student at University of Cincinnati, I saw a researcher give a talk on the pancreas, and I became very interested in the pancreas, how it functions, why it functions, what it does in the body, and what happens when pancreatic cancer arises. And I actually just cold emailed him and asked if I could join his lab as a postdoctoral research fellow. And through him, I met Marina Pasquina-Magliano at University of Michigan, who was working very actively on pancreatic cancer. And she really introduced me to the whole field and the community of researchers that are very dedicated and working very hard on pancreatic cancer. And you could really see the emotion in their eyes, telling you this story. And it's very impactful as a researcher to enter into this field and really have an obligation and a duty to try to do the very best that we can to understand more about this disease, to try to help those patients and family members as well. So I think it was a personal motivation. It was the scientific community. And also just talking with individuals who have been impacted by this terrible disease and wanting to wake up every day, and that's my motivation to come into the lab. So those are some of the reasons why I got into this field. So the next steps for my lab are a number of different aspects. I've spent the last 2 years sort of aggregating key resources, building this atlas of the human pancreas. So what we want to do now is expand that effort and really diversify the samples as well. So we are planning to utilize some of the patient blocks of tumors here at Henry Ford Pancreatic Cancer Center to be able to include more Black African American cases into the research community as a resource. So we have plans to not just perform the single-cell RNA sequencing assay, which was the nature of the atlas I mentioned, but really move into understanding the relationships, the spatial relationships of cells to one another. That's a simple principle, but in order to address that question, we have to leverage newer technologies. So the newer technology that we've been investing in and really branching forth into is called spatial transcriptomics. I think the other aspect is leveraging a very unique patient cohort or group of patients that we've been able to go back and get from archived, meaning past patient blocks. So I've been very interested in this particular concept for a while in cancer biology, particularly with pancreas, given the aggressiveness of this disease. And that is evolution of cancer over time in the same person. This is very rarely available from pancreatic cancer patients because of the aggressiveness of the disease, and often because most people are diagnosed metastatic. You don't really have insight into the beginning stages of that cancer. What was happening then, and then what was happening later on. So we've retrospectively gone back in time and the Henry Ford archives, and we've been able to identify a number of patients where we have the primary or initial tumor in the pancreas, and that will give you insight into why a particular person might not have responded to chemotherapy or radiation or had recurrence after surgery, which is really important because the vast majority of patients, even if they make it to surgery, end up relapsing and having disease progression within a pretty short time frame. So I think that this longitudinal tracking in tumor evolution, in my mind, is a key step towards making a significant impact to improve patient lives. If I had unlimited funding, I would implement this longitudinal tracking for every single pancreatic cancer patient built into their local hospital system, so that for every person that's diagnosed, when possible, you would have biobanked, longitudinal over time collection of blood and tissue when possible, and then utilize all of those translational clinical samples and put them into these cutting-edge, innovative multi-omics techniques that we have now, and really having a robust database that we can query with associated metadata, with imaging data, the CT data, and integrating all of that together, in one place, center by center. So with unlimited funds, I would build a multi-institutional, racially diverse, expanded, spatially aware genome atlas of the human pancreas. So we are very fortunate to receive the Sky Foundation Seed Award. We've been able to utilize that funding to look at expression of a particular gene that is important in driving immune suppression in pancreatic cancer. So really understanding why don't the immune cells do their job and go and kill the cancer cells. And the Sky Foundation Award supports efforts to do that project in a very specific way. So we are currently looking at not just the typical tumor samples that are evaluated in the field, which are predominantly from Caucasian or white people, but we are including a cohort of Black African American cases so that we can see whether expression of this immune checkpoint receptor is actually differently expressed in different racial groups within pancreatic tumors. We've been able to optimize this biomarker assay test not just in resected pancreatic cancer, which, as I mentioned, is the vast minority of patients, but we've now expanded this biomarker test into fine needle biopsy samples, which are taken at diagnosis, and also those metastatic biopsies at the later stages. So this is very exciting because the gene that we're looking at has several drugs that are now going through clinical trials from Genentech and other companies. So I think soon we will have some results of whether treatment with this immune checkpoint therapy improves outcomes and survival for pancreatic cancer patients. And I do visualize a test like this biomarker test, being able to integrate into the clinic. We've designed it in a very simple way so that you could plug it into a basic pathology lab and be able to get an answer. Does my particular patient have expression of this checkpoint, and to what degree? So this was really not possible without the support from Sky Foundation.